that is a polymerase chain reaction. It will keep on creating a new DNA. Okay. So we also call this artificial gene synthesis, right? Polymerase chain reaction is a molecular biology technique used to amplify a specific segment of DNA. Potentially, it was developed by Kerry Willis in 1983. Got it? See, we have simple. I'll explain a simple terminology. We have a piece of DNA fragment. Okay. Then add a prime, add a prime. Then prepare one complementary strand. We have only single stranded DNA. Then add a polymerase and add a prime and polymerase. Then create a strand complementary to that one. Right now, double stranded DNA is there. Later, again you uh, split the double stranded DNA into two single strands. Again, add the polymerase. Then again, polymerase means these two strands will develop two more strands complementary. Right now, the two double strands will form. Okay, and after that, again you cut it. Again you cut it, and again add the polymerase and nucleotides. So the four strands will become 8. 8 will become 16. 16 will become 32 like that. So it will amplify, the particular gene sequence multiple times. Got it? That is nothing but the gene amplification. Okay. So simple making multiple copies. We have one copy. Okay. Then add a complementary copy. Then you have two copies. Then separate the two copies and again add the complementary copies to the two strands. Then it will become two double strand. Then two double strand, break them. They will become four strands. And again, create complementary strands for the four strands. Then four double DNS will happen. Then break them. Then four double DNS means if you break, then they will become eight strands. Then again make them double. Then eight double strands will come. Break them. Then 16, like that. So you are amplifying a segment of gene or a part of gene multiple times. Got it? So PCR allows scientists to produce millions to billions of copies of a particular DNA sequence from a very small sample. We have only one small DNA segment we have or sample we have. You are multiplying it multiple times. Got it? So you are synthesizing an artificial gene, right? But only that particular copy only. You're amplifying it, right? So it is used in the medical diagnostic, genetic disease, and in the forensic science, research laboratories, cancer studies, infectious disease diagnosis, and prenatal testing. Also, we can use this one. This technique. You are just synthesizing, multiple genes. Okay, multiple genes means one segment. Multiple times you are synthesizing. You can sell it in the market also, because if somebody wants that particular probe, okay, or but a particular sequence, then you can sell it by naming what sequence it is. Okay, whether it is 80 GCC or GCC or 80 like that. So we can synthesize the sequences, right? And you can sell it in the market. What is the principle? Here, PCR works on the principle of denaturation of DNA, annealing of primers, extension of new DNA strands by DNA polymerase. Okay, simple. See this example, you will understand. These are the components we need. What components we need? We need a DNA template. Okay, DNA template, the one of desired one means the one which you are going to amplify. So the desired DNA template we want. Then we need the primer for initiation. Then we need the different nucleotides. Then a polymerase, that's called the tag polymerase. That's the reason we are calling it the polymerase chain reaction. Okay, tag polymerase, then PCR buffer. Now add all these. There is a thermal cycler. There is an instrument called the thermal cycler. Okay, so add all this in the thermal cycler. So what the thermal cycler do? First, it will denature the double stranded into two separate strands, right? So what the thermal cycler do? Thermal cycler will maintain the temperature. So based on the temperature, it will be separating the two strands. Okay, the double strand, it separates into two strands. Then after separating two strands, then you have to adjust the temperature. So when you adjust the temperature, then the primes will bind to the priming site here. The two primes will bind here. Okay. Then after binding, again you have to adjust the temperature. When you adjust the temperature, then this tag polymerase will be added here. Added here means initially the tag polymerase will be adding here on the DNA primer site only, right? I mean here primer site, and later on, as you are adjusting the temperature, one by one nucleotide will get added here. One by one nucleotide it will get added here, and this polymerase, tag polymerase, will be moving like this by adding different nucleotides. Then it will synthesis a chain. Here also the polymerase added here, and as the polymerase is moving, then the nucleotides will get adding here. Then it will move like this. So now two strands got created. Initially we have only one strand. Then this one strand we denatured, then annealed. Then after annealing, then it got extended. Now see here, what the denaturation do? When the denaturation happen when this solution is exposed to 92 to 94 degrees, right? Okay, so means this solution is exposed to 90 to 94. Then the double-stranded DNA will get denatured into two strands. After that, then bring the temperature to 50 to 56 degrees, not 92. Then once you bring it to the 50 to 56, the primers will get added here. See, this is a primer. Then after getting added, then again you change the temperature to 72 degrees so that the nucleotides will keep on adding here. Then it will be extended like this. Got it? Then after extending, two strands will form, right? Then again you change the temperature to 92 degrees. Again these will get denatured. Again you change it to 50 to 56. Then primer will link it to this one. Again you change the temperature. So changing the temperature outside is not possible, right? 92 to 94. Then immediately, I mean this is for example, okay, 1 second. See this. Again the same, then it's a double-stranded DNA, right? So we denatured a particular part. We denatured. Now we have two strand. Then we added the primers here. Then after adding the primers, then we are adding the nucleotides, right? When you add the nucleotides, it will give two strands now, two copies. Then again, if you change the temperature, these two will be these two will be excised to two separate strands. Then it will become four copies. Then four copies will give you 8 copies. Then after 20 to 30 cycles, you will get millions and millions of copies. Got it? It's 8 copies, right? One copy, two copies, four copies, then denature, annealing, adding the strands. Denaturing, annealing, adding the strands like this. Now you will get multiple copies. Again, the same example, double-stranded DNA, denature it, making two strands. One is sense strand, the other one is the antisense strand. So you have to expose this double-stranded DNA for 1 to 3 minutes at 94 to 98 degrees centigrade temperature, only 1 to 3. Then immediately after 3 minutes, then you have to change it to another place where the temperature is 45 to 60 seconds, then that to 30 seconds to 1 minute only. So why this? To add the primers, Then after that, immediately shift it to 72 degrees centigrade. So that that to only 20 seconds, so that the extension will finish. Okay. And later, now we have two strands. Again, you move it to 94 degrees centigrade temperature. Okay, to denature these two new strands. Again, you move it. Means changing the temperature continuously. First, you change 94 and then change it to 45 and then you change it to 75. Repeat the step 1, 2, 3, 5, 20 to 30, 35 cycles will happen. So each cycle doubles the amount of DNA and grows exponentially. All right. So terminate the PCR by cooling. If you cool at 4 degrees, then automatically the PCR technique will be turned up. Because once we have, millions of copies, then you change the temperature to 4 degrees. Then the denaturation, annealing, or extension, whatever is going on, it will get stopped. Okay. So this is the PCR. So see this denaturation, annealing, extension of new strands. The target DNA is repeatedly copied through cycles of DNA and cooling. The implication follows number of DNA copies equal to 2 to the power of n. N is the number of PCR cycles. How many cycles you do based on that one. After 30 cycles, nearly 1 billion copies of DNA will be produced. Okay. Then what are the things we need? First, template DNA, right? So this is a template DNA we need. Then we need the primers. We need the polymerase. We need the DNTTVs. What is DNTTVs? Dinucleotide triphosphates, ATGC. Then we need the magnesium chloride also. There is a cofactor. We need the buffer, which maintains the pH. Then thermal cycle is an instrument. Because we cannot do this outside, right? Because suddenly shifting the temperature from 92 to 98 and 45, it's not possible for us, right? So that is the reason there is an instrument. Okay, that's called the PCR or polymerase chain thermal cycler. Sorry, this is a thermal cycler. Can you see this? This is a thermal cycler. In the thermal cycler, we have different, the places where cuvette holders, right? So loading tray, where prepared samples are placed in tiny tubes. Then temperature control, heating and temperature control. Heating happens, refrigeration happens, mechanisms inside the machine to change the temperature quickly. Okay. Then actually there is a screen. In the screen, okay, we have to adjust. Okay, like this. What adjustment we should do? 94 to 98 degrees for 2 minutes. Then later 45 to 60 degrees for 3 seconds or 30 seconds, like this. So based on the protocol, we'll be adjusting the mission. Got it? See this? Yeah, this is a mission. Here you have to manage. The first one should be 94 degrees. How many minutes? 3 minutes. Then later change it to 34 degrees, 45 seconds, like that. I mean, whatever. Okay, see 57 degrees for 45 seconds. Then change it to 72 degrees, 1. Then step 2. Okay, again you go to means again you go from here. Okay, like this. I mean, how you want to do? We have different options, right? So select the option. Okay, then this option will help you to denature first. This is a first temperature 94 to 98, 20 to 30 seconds. Annealing needs a separate temperature. Annealing will help to bind the primers. Okay, then extension will extend the new DNA synthesis by tag polymerase. Okay, so what type of tag polymerase we use? There is a Thermus aquaticus, that is a bacteria. So from that bacteria, we are extracting this tag polymerase because that is a heat stable. It survives high temperature during denaturation, works optimally at 72 degrees. That's the reason we use this tag polymerase from Thermus aquaticus. Okay, so see this. Same DNA sample, probes, nucleotides, tag polymerase, mixed buffer, PCR. Okay, put it there. Then denatured at 95. Then 55 degrees, the primes will bind primers, right? 55 degrees. Later, synthesis happened at 72 degrees by utilizing the nucleotides and denaturing, annealing, extension. Okay, like this. See this DNA of our interest. First cycle, it gives 4, then 8 to 20 to 30 cycles, you will get millions of copies. Clear? So how is the amplification? 1, 2, if it is second cycle, 4, like that. So 30 cycles, you will get more than a billion. Clear? Then we have different techniques. In the PCR also, we have different techniques. One is the conventional PCR. The second one is the reverse transcriptase PCR. We are also called RT-PCR. Then real-time PCR. Like this, we have different types. And now I think in the market, we have around some 20 to 30 types of PCR scheme. Okay. But basically, these are very important. Conventional means the one which we discussed, the conventional. Okay. Reverse transcriptase means here what we have taken, DNA we have taken, right? But reverse transcriptation means we'll be taking the RNA. We'll taking the RNA. Then on RNA, see, for example, we are taking one RNA. RNA always exists in a single strand, not double strand, right? So if you take a RNA, then make a complementary strand of DNA. Then again on that complementary strand, create one more strand. Then that will become a DNA, right? Then you see RNA converted to the complementary DNA using the reverse transcriptase enzyme. Okay, then later it will become a complete DNA-like structure, right? Then later you amplify it. Okay, you amplify it. So for what reason we use this reverse transcriptase? To find out the disease. Like during COVID, you know COVID, right? So using the RT-PCR, we confirm the COVID positive, right? So this is a COVID diagnosis we can do. Then later real-time PCR. Real-time PCR. Real-time PCR also, it's a measures DNA amplification in real time using fluorescent dyes. Okay, fluorescent dyes. So these are the 3 methods: conventional, reverse transcriptase, and RT-PCR is a reverse transcriptase. qPCR is a real-time one, and conventional PCR. Okay. Then what are the applications of this PCR? So diagnosis of genetic diseases we do, like gene mutations, chromosomal abnormalities, inherited disorders. Simple. We amplified a gene, right? Then use that gene to compare to check whether the person is having sickle cell anemia. It will work as a probe. It will work as a probe, okay, to find out different diseases, right? Then prenatal diagnosis like Down syndrome, thalassemia, cancer diagnosis, infectious disease, okay. like this. Forensic applications also like DNA fingerprinting, in organ transplantation, HLA typing, human leukocyte antigen typing, donor recipient matching. Actually, this is my topic only. I don't know. I think I didn't explain you because that day we didn't get time to explain the complete, the immunology. So we missed the immunology. So what we do once the college opens, then again I will be taking 1 or 2 hours class on the immunology. Okay, so there you can find the HLA typing and all those. Then pharmacogenomics. Okay, so these are all the different areas where we can use this PCR, polymerase chain reaction. Okay, so don't expect long question for this. It is a short question only. Okay, and at least you will get one, multiple choice question MCQ from this PCR. Okay, yeah, that's it about the PCR. I hope you understood the class well. Then we'll meet tomorrow. So tomorrow I'll be explaining some more techniques. So by tomorrow we are going to finish the complete molecular biology. Okay. Yeah, that's it. We'll meet tomorrow at 11 then. Bye. Have a nice day.